If this is your first time here, this isn't a standalone video. It's one chapter in a complete story, the entire history of evolution, from the first replicating molecules to you. Every organism we mention is your direct ancestor, not a cousin species, not something similar, your actual lineage, an unbroken chain connecting you to the origin of life. That's what makes this different from every other evolution content out there. Most of it is scattered fragments, dinosaurs here, early humans there, the Cambrian explosion somewhere else, all disconnected. But they're not disconnected. They're all chapters in the same story, your story, and you can't understand any link without seeing what came before it. Lungs don't make sense without swim bladders. Mammals don't make sense without reptiles. Each chapter builds on the last, and nothing appears out of nowhere. 4 billion years of your deep history told as one continuous thread. If that sounds interesting, Chapter 1 is in the top right corner. I'd recommend starting there. If you've been watching from the beginning, you already know what this is. Let's keep going. Our lineage now consists of small multicellular organisms with differentiated cell types, extracellular matrices, and cell signaling systems. They can coordinate behavior across hundreds of cells, but they lack the defining features that separate animals from other multicellular eukaryotes. What makes something an animal rather than just a colony of cooperating cells? Well, the answer lies in 3 interconnected innovations: specialized tissue organized into layers, coordinated movement through muscle-like cells, and nervous systems that allow rapid communication and response. These features emerged together between 800 and 600 million years ago, transforming our ancestors from simple multicellular organisms into the first true animals. The earliest animals were soft-bodied and left almost no fossils, but we can infer their structure from modern animals that retain ancient body plans, and from rare fossil sites preserving soft tissue. The first animals were probably small, a few millimeters across, and radially symmetrical, like a cylinder or sphere. They had an outer layer of cells facing the environment, and an inner layer facing a central cavity. This 2-layer organization, with an outer ectoderm and an inner endoderm, separated by a jelly-like material, created the basic animal architecture. The critical innovation was the gut, a central cavity opening to the outside where digestion occurred externally to cells. Single-celled eukaryotes engulf food and digest it inside inner membrane-bound compartments. Early multicellular colonies probably did the same, with each cell feeding itself, but a shared gut changed everything. Cells lining the inner cavity secreted digestive enzymes into the space, breaking down food items too large for any single cell to engulf. The digested nutrients were then absorbed by the lining cells and distributed to other cells in the organism. This innovation enabled larger size and specialization. Cells on the outer surface no longer needed to feed themselves directly. They received nutrients from inner cells. This freed outer cells to specialize in protection, sensing the environment, or capturing prey. Inner cells specialized in digestion and nutrient distribution. The organism became more than a sum of its cells, because different cells performed different essential functions, creating interdependence. Coordinated movement required contractile cells, the evolutionary precursors to muscles. Early versions were probably simple epithelial cells that could change shape by contracting protein fibers running through their interior. Modern animals use actin and myosin proteins that slide past each other, generating force. These proteins existed in single-celled ancestors where they enabled cell crawling and shape changes. In early animals, cells began specializing in contraction, organizing their actin and myosin into aligned bundles that generated stronger, more directed forces. When contractile cells on one side of the organism contracted simultaneously, the body bent toward that side. When cells around the circumference contracted together, the body constricted. These simple movements, bending, constricting, extending, allowed the organism to reorient itself toward food or away from harmful conditions, to pull itself along a surface, or to change shape to capture prey. Movement was slow and imprecise compared to modern animals, but it was deliberate and responsive rather than passive drifting. Coordination required communication faster than diffusing chemicals could provide. If detecting food on the left side required minutes for a chemical signal to diffuse across the organism to trigger contractile cells on the right side, the opportunity would probably pass. The solution was nerve cells, elongated cells specialized for rapid electrical signaling. The basic mechanism exploits the fact that cell membranes maintain voltage differences through ion pumps that push sodium ions out and keep potassium ions in. When a nerve cell receives a stimulus, a chemical signal from a sensory cell detecting food, for example, channels in the membrane open, allowing sodium to rush back in. This voltage change triggers adjacent channels to open, creating a wave of depolarization racing along the nerve cell's length at speeds measured in meters per second rather than millimeters per second of diffusion. Where the nerve cell contacts a contractile cell, the electrical signal triggers release of chemical neurotransmitters that bind to receptors on the contractile cell, causing it to contract. A network of nerve cells connecting sensory cells to contractile cells throughout the body allowed coordinated responses within fractions of a second. An organism detecting food on its left side could bend left almost immediately, tracking the source while it remained nearby. These three innovations, layered tissue organization with a gut, contractile cells enabling movement, and nerve cells enabling coordination, define animals. Every animal, from sponges to humans, possess some version of these features. Sponges are the exception that proves the rule. They have differentiated cell types and coordinated feeding currents, but they lack true nerves and muscles and cannot move as whole organisms. Most biologists consider sponges animals, but the earliest branching animals, representing the baseline from which everything else elaborated. Our lineage moved past the sponge-grade organization toward more complex animals called metazoans, animals with true tissues, nerves, and muscles. The earliest metazoans lived between 650 and 600 million years ago, during a period of dramatic environmental change. Earth had just emerged from a severe glaciation period, where ice sheets extended nearly to the equator. As the planet warmed and ice retreated, ocean chemistry changed, oxygen levels rose further, and new ecological opportunities opened. These early metazoans resembled modern cnidarians. They had radial symmetry, meaning their bodies were organized around a central axis, like spokes on a wheel. They had tentacles surrounding a mouth opening into a central gut cavity. The tentacles, armed with specialized stinging cells, captured small prey and pushed them into the mouth. Nerve cells form a loose network throughout the body, allowing coordinated tentacle movements and body contractions. The Ediacaran period, from 635 to 541 million years ago, saw these early animals diversify into a variety of body forms, preserved as fossils in a few exceptional sites. The fossils show disc-shaped organisms, frond-like organisms anchored to the seafloor, and segmentic worm-like organisms. Many of these are difficult to interpret. They don't fit cleanly into modern animal groups, and some might represent extinct experimental body plans that left no descendants. What's clear is that by 550 million years ago, animals had spread across the sea floor in shallow marine environments. They remained relatively simple. Soft-bodied, small, probably slow-moving. No hard shells, no skeletons, or complex organs. But they were unquestionably animals. Multicellular organisms with differentiated tissues, guts, nerves, and muscles, actively moving and feeding on other organisms or filtering particles from water. The constraint limiting these early animals was predation, or rather, the lack of it. Without significant predators, there was little pressure to evolve defenses like shells or escape behaviors like rapid swimming. Without active predators, there was less advantage to evolving sophisticated sensory systems or powerful muscles. The Ediacaran fauna existed in a relatively peaceful world where competition for space and food mattered more than avoiding being eaten. This changed at the Ediacaran-Cambrian boundary around 541 million years ago. The fossil record shows a sudden shift. Ediacaran-style organisms disappear, and new animals forms with hard shells, mineralized skeletons, and evidence of predation appear. This transition marks the beginning of the Cambrian explosion. But before we get there, we need to identify where our specific lineage was during the Ediacaran. Our ancestors were probably small, soft-bodied, worm-like organisms living on or in seafloor sediments. They weren't the large frond-like fossils that dominate some Ediacaran sites. They were bilaterans, animals with bilateral symmetry, meaning their bodies had distinct left and right sides, a front and back, and a top and bottom. This body plan, which seemed obvious for any mobile animal today, was an innovation during this period. Bilateral symmetry makes sense for directed movement. A radially symmetric animal, like a jellyfish, moves vertically or drifts with currents. It has no preferred horizontal direction. A bilaterally symmetrical animal moves forward with sensory organs concentrated at the front to detect what it's approaching, a mouth positioned to consume what it encounters, and propulsive structures pushing from behind. This organization creates a head-tail axis and a dorsal-ventricle axis, fundamentally restructuring the body compared to radial ancestors. The genetic mechanisms enabling bilateral symmetry involve Hox genes, regulatory genes that specify position along the head-tail axis. These genes existed in earlier animals, but became elaborated in bilaterans into a coordinated system that divided the body into distinct regions, each with different structures. Hox genes act as master switches, activating different sets of downstream genes in different body regions. The front expresses certain Hox genes and develops head structures. The middle expresses different combinations and develops trunk structures. The rear develops tail structures. Early bilaterans were likely small and worm-like because this body plan suits burrowing. As animals evolved to exploit sediments for food and shelter, bilateral symmetry and elongated bodies became advantageous. An organism could push its front end forward through sediment, using muscles to generate sequential waves of contraction traveling from head to tail, propelling itself efficiently through a dense medium. Our specific ancestors during the late Ediacaran were probably coelomates, bilaterans lacking a body cavity between the gut and outer body wall. Their insides were solid tissue except for the central gut cavity. This limited their size because nutrients and oxygen had to diffuse through tissue from the gut and outer surface to reach internal cells, but it was mechanically simpler and sufficient for small organisms. By the end of the Ediacaran, around 541 million years ago, our lineage consisted of small, bilaterally symmetrical, soft-bodied worms a few millimeters to perhaps a centimeter long. They had a through gut, a mouth at one end and an anus at the other, allowing one-way flow of food and more efficient digestion than the single-opening guts of cnidarians. They had longitudinal and circular muscles, allowing sophisticated crawling and burrowing. They had a concentration of nerve cells at the front end, forming a simple brain, coordinating sensory input and motor output. They likely had simple, light-sensitive cells detecting direction and intensity but not forming images. These animals weren't impressive by modern standards. They'd be pretty easy to overlook in a modern tidal pool, but they possessed the fundamental bilateran body plan that every animal in our lineage since has elaborated. The head-tail axis, the left-right symmetry, the dorsal-ventral organization, the through gut, the anterior nerve concentration, these features, established during the Ediacaran, remains visible in every vertebrate, including humans. The stage was now set for the most dramatic diversification in animal history. Oxygen levels continued rising, approaching modern concentrations. Ecological relationships intensified as animals evolved to prey on each other more effectively. And crucially, animals evolved biomineralization, the ability to construct hard structures from calcium carbonate, calcium phosphate, and silica. Hard parts enabled new defenses, new modes of predation, new body support systems, and crucially for paleontologists, a fossil record that suddenly becomes rich and detailed. The explosion of animal diversity preserved in that fossil record and our lineage's role in it is the next chapter.